The Muscular System, Chapter 6, Part 1. Because flexing muscles look like mice scurrying beneath the skin, some scientists long ago dubbed them muscles, from the Latin word mus, meaning little mouse. Indeed, the rippling muscles of professional boxers or weightlifters are often the first thing that comes to mind when someone hears the word muscle. But muscle is also the dominant tissue in the heart and in the walls of other hollow organs of the body. In all its forms, it makes up nearly half the body's mass. The essential function of muscle is contraction or shortening, a unique characteristic that sets it apart from any other body tissue. As a result of this ability, muscles are responsible for essentially all body movement and can be viewed as the machines of the body. There are three types of muscle tissue: skeletal, cardiac, and smooth. These differ in their cell structure, body location, and how they are stimulated to contract. But before we explore their differences, let's look at some of the ways that they are the same. First, skeletal and smooth muscle cells are elongated. For this reason, these types of muscle cells are called muscle fibers. Second, the ability of a muscle to shorten or contract depends on two types of myofilaments, the muscle cell equivalents of the microfilaments of the cytoskeleton. A third similarity has to do with terminology. Whenever you see the prefixes myo, M-Y-O, or M-Y-S, or sarco, you will know that muscle is being referred to. For example, in muscle cells, the cytoplasm is called sarcoplasm. Here is a comparison of the three types of muscles. And here you can see the regulation, the speed, and the rhythm of contraction. Skeletal muscle fibers are packaged into the organs called skeletal muscles that attach to the body's skeleton. Skeletal muscle fibers are huge, cigar-shaped, multinucleated cells. They are the largest of the muscle fiber types, some ranging up to 30 centimeters. That's almost a foot in length. Indeed, the fibers of large heart-working muscles, such as the anti-gravity muscles of the hip, are so big and coarse that they can be seen with the naked eye. Skeletal muscle is also known as striated muscle because its fibers have obvious stripes, and as voluntary muscle because it is the only muscle type subject to conscious control. However, it is important to recognize that skeletal muscles are often activated by reflexes without our willed command as well. When you think of skeletal muscle tissue, the key words to remember are skeletal, striated, and voluntary. Skeletal muscle tissue can rapidly contract and with great force, but it tires easily and must rest after short periods of activity. Each muscle fiber is enclosed in a delicate connective tissue sheath called endomysium. Several sheathed muscle fibers are then wrapped by a coarser fibrous membrane called a perimysium to form a bundle of fibers called a fascicle. Many fascicles are bound together by an even tougher overcoat of connective tissue called an epimysium, which covers the entire muscle. Here you can see the different layers of skeletal muscle. The epimysia blend either into strong cord-like tendons or into sheet-like aponeuroses, which attach muscles indirectly to bones, cartilages, connective tissue coverings. Producing movement is a common function of all muscle types, but skeletal muscle plays three other important roles in the body as well. It maintains posture, stabilizes joints, and generates heat. As mentioned previously, skeletal muscle cells are multinucleated. Many oval nuclei can be seen just beneath the plasma membrane, which is called the sarcolemma in muscle cells. The nuclei are pushed aside by long ribbon-like organelles, the myofibrils. Another very important muscle fiber organelle, the sarcoplasmic reticulum, is a specialized smooth endoplasmic reticulum. The interconnecting tubules and sacs of the SR surround each and every myofibril, just as the sleeve of a loosely crocheted sweater surrounds your arm. This shows a portion of a muscle fiber. One myofibril has been extended for you to look at. Alternating light and dark bands along the length of the perfectly aligned myofibrils give the muscle cell as a whole its striped appearance. Think of the second letter of light, I, and the second letter of dark, A, to help you remember which band is which. A closer look at the banding pattern reveals that the light I band has a midline interruption, or darker area called the Z disk, and the dark A band has a lighter central area called the H zone. The M line is the center of the H zone, contains tiny protein rods that hold adjacent thick filaments together. Here you can see the enlarged view of a section of a myofibril showing you the banding pattern. So why are we bothering with all these terms, dark this and light that? Because the banding pattern reveals the working structure of the myofibrils. First, we find that the myofibrils are actually chains of tiny contractile units called sarcomeres, which are aligned end to end like boxcars in a train along the length of the myofibril. Now, second, it is the arrangement of even smaller structures, these myofilaments within the sarcomere, that actually produce the banding pattern. There are two types of thread-like protein myofilaments within each of our boxcar sarcomeres. The larger thick filaments, also called myosin filaments, and the thin filaments are composed of the contractile protein called actin. Now, as I said, the larger thick filaments are made mostly of bundled molecules of the protein myosin, but they also contain ATPase enzymes, which split ATP to generate the power for muscle contraction. Notice that the thick filaments extend the entire length of the dark A band. Also notice that the mid parts of the thick filaments are smooth, but their ends are stubbed with small projections. These projections, or myosin heads, are called cross bridges when they link the thick and the thin filaments together during contraction. The thin filaments are composed of the contractile protein called actin, plus some regulatory proteins that play a role in allowing or preventing binding of myosin heads to actin. The thin filaments, also called actin filaments, are anchored to the Z disc, which is a disc-like membrane. Here you can see the structure of the thick and thin filaments found in the sarcomeres. Although they overlap the ends of the thick filaments, the thin filaments do not extend into the middle of a relaxed sarcomere, and thus the central region, which is called the H zone, looks a bit lighter. The major role of the sarcoplasmic reticulum is to store calcium and to release it on demand when the muscle fiber is stimulated to contract. As you will see, calcium provides the final go signal for contraction. This diagram shows the structure of the thick and thin filaments found in the sarcomeres. So what did we learn here in this particular lecture? Here are the key points. How do cells of the three types of muscle tissues differ from one another anatomically? Which muscle type has the most elaborate connective tissue wrappings? What does started mean relative to muscle cells, or striated is what that's supposed to be? And specifically, what is responsible for the banding pattern in skeletal muscle cells? This ends lecture number 1. We're going to go on to lecture number 2.